this afternoon Tony Planchart, who's going to be giving his seminar. Tony is being considered for promotion to associate with tenure. Tony received his PhD in molecular biology and biophysics from Vanderbilt, and then went on to do a postdoc in biophysics at Harvard. His second postdoc in mammalian genetics at the Jackson Lab in Bar Harbor, Maine. Apparently liked it in Maine because then he hung out there, ultimately settling in at Mount Desert Island Biological Labs, where he had the position of assistant professor. That's where he was and what he was doing when we scooped him up in 2012 and came to the department as an assistant professor. Tony is a strong advocate of the use of the zebrafish model in biological research, both here within the state, but he's also pretty much internationally recognized for his work with the zebrafish model and in promoting the zebrafish model. Here, he was instrumental in securing funding for the establishment of the zebrafish core facility, which is over in the toxicology building. And he also chaired a major international conference on the use of the zebrafish and other small aquarium fish as biological models. He obviously uses the zebrafish currently in his own toxicological research. And the title of his talk today is Living in a Toxic Fishbowl: How Zebrafish Are Accelerating Mechanistic Insights into the Origin of Human Disease.
Thank you, Jerry. Thank you for that kind introduction. You pretty much covered half my seminar. So thank you all for coming. I really appreciate it. It's nice to see friendly faces that I don't often encounter because I'm not over here as much as I used to be. And if you will bear with me 1 second, I will have my iPhone ready to go.
It's very difficult to hear you back here.
I'm sorry. How's that? Is that better?
Yes.
Okay. I will speak up. Okay, so as Jerry said, that's the title of my talk. And I have 4 objectives today. I'm going to tell you 3 short stories, and then I want to tell you a little bit about what I've been doing the rest of the time. So you'll see this slide recur again several times. So if you don't have a chance to read it all now, don't worry about it. But we're basically going to be talking about pharmaceuticals and neurological impairment, heavy metals in our environment, and balance and coordination problems, which we think could have application to ADHD. And I'm going to tell you a cautionary tale on the use of antisense technologies, in particular morpholinos, that I have experienced in my own lab, although we think we can still salvage a lot of the data. So as Jerry said, I got my PhD at Vanderbilt, but really it all started right here in Oklahoma, where I was born. And then I was flown to Venezuela, where I was raised for about 15 years. Came back to Texas and spent a few years there going to Texas A&M. They have a canine mascot, although it's almost always a mutt. Vanderbilt, followed by Boston, and then spent about 15 years in Maine, where I worked as a postdoc in the Jackson Laboratory on mammalian genetics. While at Harvard, I caught the genome bug. It was a time when ESTs were being dumped in immense numbers into the DB EST, and people were using this as a mapping reagent. It was revolutionizing how people were mapping genes. And so it seemed to me logical to go to the Jackson Laboratory and instead of stay as a crystallographer, which would eventually be robotized, which it now is, I could gain invaluable experience as a molecular geneticist working with mice. While there, I taught at the College of the Atlantic and Bates College as well for a couple of years, and then ended up at MBI Biolabs, where I started the first zebrafish facility. So what is this title about? I hope to convince you that we all start life in a fishbowl. Because today is the 50th anniversary of the airing of the first episode of Star Trek, I thought we would start by going boldly in that direction and seeing what we have in terms of this idea of living in a fishbowl. So here we are on planet Earth. It's mostly water with a few rocks floating about where we exist. And we're right about here where that asterisk is. I'm sure it's much sunnier now than it was then. But this picture belies a more unsettling reality, and that is that we are causing significant destruction to our ecosystem. Now, we live in this nice green patch of the Western industrialized world, but everything that we consume in terms of electronics and other products is made somewhere else where there's a lot of nasty things going on. And these things naturally become in equilibrium with our ecosystem, and I would posit eventually affect us. And the way they do that is oftentimes embryonically. So if you look at this image of a human embryo, this is the amniotic sac, and that embryo is developing within the amniotic fluid. So it is, by definition, something of an aquatic organism at this stage of its life. And the troubling thing about it is, if I can work, is that everything that we come into contact with, if the mother comes into contact with any of these things, including industrial waste, personal habits, pesticides, recreational drugs, heavy metals, cosmetics, pharmaceuticals, and things intended to make us clean and germ-free, eventually make their way into the amniotic fluid. And this has been shown. People have sampled amniotic fluid, they have subjected it to analysis, and they have found all sorts of nasty and non-innocuous things within the amniotic fluid. So our exposure starts very early in life. And so what better way to study an aquatic-based exposure than to use a tractable aquatic model organism such as the zebrafish? Now, I can't talk about all of these things today, so I'm just going to focus on two, heavy metals and pharmaceuticals. And the thing to keep in mind is that if we don't do something about this problem with our environment, it's only going to come back to affect us all in some significant way, whether now or later down the road. And despite all the recent exoplanet discoveries and nearby stars and the idea that we can go to Mars and colonize it, good luck with that. I don't think that's necessarily going to happen in any of our lifetimes. And it's better to keep in mind Gary Larson's advice that, yeah, you got out, but we're still screwed, okay? So we should probably try to do something about it. And one thing we can do is try to understand how these environmental exposures affect us all at the embryonic level and predispose us for disease later in life. And so that's why I use zebrafish, or Danio rerio. They're not that big, they're actually quite small. If you have ever had an aquarium, could I see a show of hands? Who here has ever had zebrafish in their aquariums? It's a very common, very popular pet. And I have a bunch at home, and since this is being recorded, I want the dean and the IACUC to know that I did not take those from my fish facility. I actually bought them at a pet store. Now that that's on the record, we can move on. So why zebrafish? Well, the first and most obvious reason is that it's a vertebrate. So just like us, it undergoes the same fundamental organogenic programs that we do, with some exceptions. They don't make a respiratory system like we do. It is extremely fecund. We can get about 200 eggs from a female per week, and that helps us in our research. It has a rapid developmental time point from embryo, from one cell stage embryo, to a free-swimming larvae that is close to feeding, approximately 96 hours. It's experimentally tractable, meaning that we can manipulate it. We can manipulate its genome. We can use it for exposure science. We can use it for developmental biology. And we can do it in ways that are, I would posit, much easier than most other vertebrate model organisms, simply because they develop externally instead of developing in utero. And as you probably become aware, there are a large number of reagents available for use with zebrafish, not the least of which is the new CRISPR craze. but we also have large numbers of transgenics, and we have large numbers of probes that we can use. But perhaps more importantly is that they share evolutionary kinship with us. And so this circus diagram right here illustrates that point. And for those of you not familiar with these diagrams, each of these colored blocks on the human side is a chromosome, chromosome 1 through 23, and then the X chromosome. And what the author here has done is taken all the genes that are synthetic, that is within one chromosome, and shown how they have been dispersed throughout evolution on these other model organisms. And of course, our closest kinship illustrates that we have significant conservation of synteny between us and the chimps. But as we move away down the evolutionary tree, these genes become more dispersed, and synteny is broken, but nevertheless, they're still there. And oftentimes, they're functioning in the exact same pathways in which they function in humans. And so it makes it possible for us then to ask the question of what happens when I perturb a genetic pathway in this organism and gain valuable insight into what might be happening in this organism, which we care considerably about, especially if you're the parent of that beautiful child. The other thing, too, as Jerry alluded to, is that we have this awesome zebrafish facility, which was built, and I'll tell you more about that later on, in the toxicology basement. It is a state-of-the-art zebrafish facility with a very sophisticated water purification system. It even has a thing called the UV reactor to purify water. It can house 20 to 30,000 fish in recirculating racks. And we also have the ability to generate over 1,000 embryos once this thing is online, because we can house over 100 fish in there to spawn them on a daily basis and get 1,000 eggs a day. And then we also have standalone racks in that room for exposure science that we might be wanting to do to keep them separate from our stock fish. In addition to this major facility, we also have a quarantine room with 2 standalone racks. And if you haven't seen the facility, come on over. We're happy to give you a tour. So I hope I've convinced you that there are genuinely logical reasons by which one would want to study certain aspects of environmental science and developmental biology in zebrafish. And now I hope to expand upon that. So I'm going to give you the first story, which is this drug known as leflunomide, which is a disease-modifying anti-rheumatic drug, or DMARD. It is a potent teratogen in many model organisms, but it is one of the first lines of defense against rheumatoid arthritis. So when people take this, particularly women, they are told explicitly not to get pregnant, and if they risk getting pregnant, that they must go off of this drug and engage in a regimen of charcoal purification to remove any residual traces. It takes approximately 11 to 12 months for this drug to be undetectable in the bloodstream after you've gone off of it. So it's a profoundly lasting drug. And we have data to suggest that it could potentially cause neurological impairments. And I hope to show you, or I hope to convince you of that fact now. Oh, there we go. So it is an immunosuppressive. It's not only used to treat rheumatoid arthritis, but also MS and lupus. Its active metabolite inhibits an enzyme called dihydroorotate dehydrogenase, which is the rate-limiting enzyme in the de novo pyrimidine biosynthesis pathway. And this is important because T cells, which are the ones that are responsible for the inflammatory phenotype in rheumatoid arthritis, use the de novo pyrimidine biosynthesis pathway almost exclusively and don't avail themselves of the salvage pathway. So if you deny them the ability to make pyrimidines de novo, then what you do is you dampen their ability to reproduce when they are called to do so. It's also known to inhibit melanin synthesis in zebrafish, and it affects to some degree neuropressed cells and their derivatives, but at very high concentrations, concentrations that would not be therapeutic by nature. But because of that, it is now being eyed as a potential treatment for melanoma. There are clinical trials underway using this drug to treat stage 4 melanoma. It is also a fairly potent aryl hydrocarbon receptor ligand. And for those of you that are not familiar with the aryl hydrocarbon receptor pathway, it is a classical signal transduction pathway. The prototype ligand is known as 2,3,7,8 tetrachlorodibenzoparadioxin, or TCDD, often referred to in the literature as dioxin. And this particular ligand binds to the aryl hydrocarbon receptor at sub-nanomolar affinities, so it's a very potent activator of the receptor. Now, under ordinary circumstances, the aryl hydrocarbon receptor pathway is not active because in the absence of ligand, the aryl hydrocarbon receptor is sequestered in the cytoplasm by chaperonin proteins. But upon a ligand diffusing through the plasma membrane, the aryl hydrocarbon receptor dissociates from the chaperonins, allowing it then to enter the nucleus, where it is an obligate heterodimer with another nuclear protein known as the aryl hydrocarbon receptor nuclear translocator. These two function as a competent transcription factor, and they recognize specific elements within the DNA known as aryl hydrocarbon response elements and activate transcription of a battery of genes. Now, typically these genes are genes involved in xenobiotic metabolism, so it includes a lot of the P450 cytochrome proteins, but they also activate a number of other genes that are not necessarily involved in the xenobiotic process, but are involved in other aspects of development and potentially health. So now that you know about the aryl hydrocarbon receptor, the structure of leflunomide looks nothing like the prototypical ligand, but it nevertheless still binds. So this is a very promiscuous receptor, and it's something that Seth Coleman and I have thought about investigating in terms of non-classical ligands and looking at how they bind and activate the aryl hydrocarbon receptor pathway, and I'll hopefully touch on that later on. But for leflunomide to actually be an anti-rheumatic drug, it needs to be converted to teriflunomide, and that's typically done by the cytochrome P450 family, albeit not those members of the family that are activated by the aryl hydrocarbon receptor family. And so when we expose zebrafish to either DMSO or teriflunomide, we see no effect on the zebrafish. In fact, you see nice melanocytes, fairly decent development. But if you expose it to a similar concentration of leflunomide, the precursor of the active compound, you see a complete abolishment of melanocytes. And so that raised the question for us, well, you know, what pathway is involved in melanin synthesis? And hearkening back to my biochemistry was not going to be the solution to that, because I couldn't remember exactly what the derivative, what melanin was derived from. So I had to look up the pathway. And what we see here is that melanin is a derivative of tyrosine, but then again, so is dopamine. And in fact, they share common enzymes, tyrosinase and tyrosine hydroxylase. Tyrosinase is primarily involved in the production of dopaquinone, which is a precursor of melanin, whereas tyrosine hydroxylase is involved in the production of L-dopa, which is a precursor of dopamine. And so the pathway from tyrosine to dopamine is just a two-enzyme step, tyrosine hydroxylase and dopamine decarboxylase. There is some evidence in the literature that tyrosinase might actually play a role in this branch of the pathway or interconvert these two, but the evidence for that is relatively weak. So if in fact this melanin pathway is affected, as we can see by the phenotype of the fish, what effect is this compound having on the gene expression profile of some of the enzymes involved in this pathway? And so here you can see that when we expose these zebrafish to leflunomide, we get a significant downregulation of tyrosinase. So that's consistent with the melanin phenotype that we observe. But more importantly, we also see a significant downregulation of tyrosine hydroxylase. Now, we don't have a handle yet on why this is, because it doesn't seem that this downregulation is a direct effect by the ligand-bound aryl hydrocarbon receptor. There's probably an intermediate in that pathway. But as I'll show you, we know that the aryl hydrocarbon receptor appears to be necessary for this to happen. And then we followed up the observation of tyrosine hydroxylase downregulation by looking at where this downregulation is occurring in the zebrafish. And so these, excuse me, these are zebrafish embryos. I think they're about 36 hours old. And you can see here that the tyrosine hydroxylase is positively labeling a cluster of cells in the developing central nervous system of the zebrafish brain. But that same cluster has some spatial alterations in the expression profile of tyrosine hydroxylase in the leflunomide-exposed zebrafish. Now, not being neuroanatomists, we weren't exactly sure what we were looking at, but we took advantage of the fact that we had a transgenic zebrafish in the lab, the so-called DAP-GFP, which expresses the dopamine, GFP under the control of the dopamine transporter, and labels those cells that are dopaminergic in nature. And so you can see here that there are a number of cells in the zebrafish central nervous system that express this dopaminergic transporter by proxy in terms of looking at GFP expression. So you have the olfactory bulb, we have the forebrain region, the midbrain region, and the hindbrain region with the midbrain, hindbrain boundary here. So this is the nose of the zebrafish, and the tail would be in this direction. And absent from this expression profile were a cluster of cells that correspond to the midbrain region, which is exactly where you would expect dopaminergic cells to be located. Now, the zebrafish don't have a substantia nigra like we do, which is where our dopaminergic neurons reside. But they do nevertheless have a defined cluster of dopaminergic cells in this region of the brain corresponding to what would be, in a human, the midbrain region and the ensuing substantia nigra. Furthermore, we went on to show that there are actually, there's a diminishment in dopamine in the brains of these zebrafish. So in collaboration with a colleague, Jason Richardson, we looked at the dopamine metabolic pathways. So here you can see that dopamine is metabolized to DOPAC by these monoamine oxidases, and then by COMT to homovanillic acid. And when we look at these different compounds, dopamine, DOPAC, and homovanillic acid, what we see is that in a concentration-dependent manner, we have a substantial decrease in dopamine in the brain of these zebrafish. And DOPAC, on the other hand, does not seem to be affected, which suggests that there's maybe some negative feedback on this enzyme when there's not enough dopamine around. Whereas homovanillic acid, the endpoint of this metabolic pathway, is completely absent from these fish, suggesting that these cells are doing everything they can to keep dopamine around. Now, we thought we would look at some of the dopamine transporters, but so far we haven't seen any changes in their expression, at least at the gene level. But we don't have yet the probes to analyze whether or not the protein channels are still there, or if their recycling properties have changed, but that's something that we are looking at. So if the dopamine system seems to be affected, then one would expect that there would be a behavioral effect as well. And so we use the Nolvis apparatus and EthoVision software, which we purchased recently through a grant, to look at the behavior of these zebrafish. So in green we have, in red we have the leflunomide exposed zebrafish. In green we have the teriflunomide, which is the active metabolite in rheumatoid arthritis. And then in blue we have a vehicle control. And the way to interpret this experiment is that zebrafish are extremely active in the dark and not so much in the light, so that we can take advantage of that property and we can control when the light comes on and when it goes off, and we can measure the amount of movement within an arena and then integrate that movement and get an idea about how active they are within that period of time. So wherever you see these light-shaded gray bars, that's when we turn the lights off, where you can see immediately that the activity of these zebrafish goes up in a regular, repeating way after a short period of acclimation in the system. And so we see here that the wild type and the teriflunomide track together. In other words, teriflunomide, we already knew, didn't have this effect on melanin. It also doesn't have the effect on tyrosine hydroxylase, nor does it have the effect on dopamine. So we would expect it to track. But more significantly, the leflunomide movement is substantially depressed. In other words, these fish are not moving nearly as much in these arenas. And that is a typical indication of a problem with the dopaminergic system. Your movements tend to be slightly more erratic. You tend to be much more static than you are if you don't have this loss of dopaminergic neurons. And this is important because, well, the principal disease associated with the loss of dopaminergic neurons is Parkinson's disease, right? And so we are trying to ask the question, does exposure to these compounds that affect the dopaminergic system in these zebrafish embryos translate to potential risk in individuals over time in acquiring Parkinson-like symptoms? Will we see Parkinson-like symptoms go up in people who are treated with these types of compounds? Now, the aryl hydrocarbon receptor seems to mediate to some degree that phenotype that we see, because when we take a knockout of the aryl hydrocarbon receptor, and zebrafish have 3 because of a genome duplication that took place several million years ago. They have 1A, 1B, and 2, whereas we just have 1. But AHR2 seems to be the most dominant in the signal transduction pathway of the aryl hydrocarbon receptor. So when we knock it out, we see that in fact now all 3 track together. So this is an indication that at least this phenotype seems to be in part mediated by the aryl hydrocarbon receptor pathway. Now you might ask yourself, well, is there a potential problem looking at embryos in which perhaps the blood-brain barrier has not fully developed? And this is a legitimate concern. In zebrafish, the blood-brain barrier doesn't develop for at least 3 to 4 days after fertilization, during the larval stage. So to get to that question, we said, well, let's see what happens in adults. But before I move on to that, sorry, I got ahead of myself. We also see a variation in the susceptibility to leflunomide. So if you take a group of embryos derived from the same spawning event and you expose them to leflunomide, 500 nanomole, you will get a clutch of these embryos that are highly susceptible in terms of their inability to synthesize melanin, and others that look relatively indistinguishable. And if you raise them out, you will see that you will have a significant number of these embryos that have adverse developmental phenotypes. Whereas the ones that are somewhat resistant look relatively normal, pencil-shaped zebrafish. They're starting to use up their yolk. And what we don't yet have a handle on is what is the genetic basis for this? Is the genetic basis rooted in the embryo's ability to resist the effects of leflunomide, or is the genetic basis rooted in the construction of the chorion that surrounds the embryo? Is there a structural barrier difference between the chorion of susceptible zebrafish and non-susceptible zebrafish? And so we're working to address that question as well. Okay, so back to the question about the blood-brain barrier. So, not sure how much you know about neurovascular units. I had to actually read up quite a bit on this. But it turns out that the blood-brain barrier is entirely the responsibility of the endothelial cells that line the blood capillaries in the brain. They happen to have significant numbers of tight junctions that are only permeable to small ions and gases. And then there are other structural supports. So you've got pericytes, which are contractile cells, and you've got astrocytes that pretty much wrap around the vessel. But the entire blood-brain barrier depends on those tight junctions within endothelial cells. And that's a distinction between peripheral capillaries and central nervous system capillaries. Our capillaries in our periphery are quite leaky, but the capillaries in the brain are not. This also provides a significantly powerful barrier to the ingression of compounds from the blood into the brain. And there are a significant number of ABC transporters within these cells that actively pump things out and back into the blood, not letting them get in. So it's a legitimate question if this compound is affecting the behavior of larval zebrafish who have not quite yet cemented their blood-brain barrier. Is that really significant for adults? And so we wanted to address that question. And what we did was take advantage of the novel tank assay. By the way, I forgot to mention that most of this work was done, if not all, by Elizabeth Cook, who is sitting right here, one of my graduate students. And she devoted hours and hours and hours to these measurements. So the novel tank assay simply asks, when you take a zebrafish from its original tank and you put it in a novel setting, it typically will dive to the bottom as a response to avoiding predatory capture. And it will swim in the bottom for a while. And as its anxiety ameliorates, it will begin to swim up and explore the region of the tank to a greater degree. So we can subdivide a novel tank into bottom zones and top zones, and then when we put the fish in, we can ask, how long does it spend in the bottom before it starts to explore its tank? And get a measure of the zebrafish's anxiety or exploratory curiosity by making those measurements. So I'm going to show you two short videos where the zebrafish have been dropped in the tank, and I want you to guess by looking at these which one is the control and which one is the leflunomide exposed zebrafish. Now keep in mind the characteristics of an anxious fish are jittery swimming, freezing, very little exploration of its environment, whereas a less anxious fish will swim throughout the water column once it becomes acclimated to its tank in a short period of time. So have you made up your mind which one is the control and which one is the experimental?
Top one's the control.
There you go. Top one's the control. Exactly. And when we integrate that behavior and display it as a heat map, you can see right away how pronounced the amount of motion within that tank is in a control relative to a leflunomide exposed adult. So this is clearly indicating that this drug is having a behavioral effect on these adult zebrafish and suggests that it can get past the blood-brain barrier. Something that you would be surprised to learn, no published literature exists on whether or not this stuff crosses the blood-brain barrier in humans. I guess because there aren't many people who will volunteer for cerebral spinal taps to analyze these things. And then when we quantify it, you can see that the zebrafish that are exposed to the vehicle control, their mean distance to the bottom zone is significantly greater than the mean distance to the bottom zone of the leflunomide exposed zebrafish. And when we look at the time domain, we can see that their cumulative duration in the bottom of the tank is significantly shorter, whereas the leflunomide anxious fish spend a much greater amount of time in it. So other things that we are investigating, and we're on the cusp of writing this up and submitting it for publication, but there are still a few things that we would like to know. We'd like to know what the degree of heritability of this resistance is and whether or not it's a structural or an embryonic property of the zebrafish. We would like to know if leflunomide promotes an inflammatory response in the central nervous system, because that could also explain some of the behavioral observations we're making. And we would like to know if there are long-term behavioral effects due to some neuronal wiring that occurs early in the zebrafish's life that can be propagated to later in life. So we're doing some longitudinal studies on that as well. So that's the short story number one. The short story number two is about how heavy metals in our environment cause balance and coordination problems in children. Now, this work has largely been done by Adrian Green, another graduate student in the lab who's sitting here. And through conversations with Catherine Hoyo, our colleague, we learned that children with high urinary and blood levels of heavy metals, including lead and cadmium, are at higher risk of developing attention deficit hyperactivity disorder. And through reading the literature, we also found that a significant number of these children also exhibit coordination and balance problems, even though no one has actually investigated why that is. So we thought, well, can we apply our zebrafish model to study this effect? Well, as I'm sure all of you know, the balance and coordination are regulated by the inner ear. And the developing inner ear of a zebrafish ends up, as an adult, very similar to the developed inner ear of a mammalian organism, including the production of semicircular canals, utricles and saccules, and the lagina, which becomes the cochlea in higher mammals, or in mammals in general, is also a source of sound integration for zebrafish. Although the way they integrate sound is a lot different than the way we integrate sound, because they don't have, excuse me, a middle ear per se. They rely actually on their air bladder, their swim bladder, to do that through a very complex system called the Weberian apparatus. But the point is that balance and coordination are regulated within the utricle and saccule and the lagina in zebrafish by the presence of these mineraloprotein aggregates that we call otoliths in zebrafish, and human biologists call otoconia in humans. And these are present in three compartments in the zebrafish: one in the utricle, one in the saccule, and one in the lagina. And when you look at their relative sizes, the saccule otolith is significantly larger, rounder in shape. The one in the utricle is ellipsoid, and the lagina is actually known as an asteriscus because it's so tiny. But when you look at it from a structural standpoint, these crystals are present in larger numbers in the human or mammalian inner ear. And they're faceted crystals, and they rest upon a ciliated cell mat. And it is within the endolymph of the ear. And as you move side to side, or you accelerate, these little stones move around in your inner ear, and you integrate that information as acceleration, as movement. And if something goes awry, you have balance and orientation disorders. And so over here you can see, very similarly, these otoliths sit, although the resolution is not that great, on ciliated hair cells as well. And whenever the fish moves, these things move as well, and that gives them the same ability to integrate spatial information in the brain. So we did a really simple experiment. We exposed zebrafish to cadmium and asked what happens to various elements of the skeleton and the inner ear. And as you can see right here in the control, we have very nicely formed otoliths. You can see them both dorsally and then flip the fish around ventrally. But on the cadmium side, they're completely gone. Now, I should tell you that it's not that they didn't form to begin with. The process of clarifying these zebrafish involves a trypsin step. And so what's happened is that they've broken down, and they're no longer there. So it suggests that the protein composition of the otoliths in these cadmium-exposed zebrafish is different in some way than the otolith composition of the non-exposed zebrafish. So one of the things that we've learned from looking at otolith synthesis or biosynthesis is that otoliths are high in calcium carbonate. And the endolymph of the ear is very low in calcium. There's actually a number of channels that are busy pumping calcium into this otic space in order for there to be enough to nucleate crystals of these organic protein crystal matrices. So we asked if we could rescue this phenotype. Oh, I'm sorry, this is the behavior. So we asked if, in fact, the loss of these otoliths in cadmium affects their behavior. And so we have various ways of measuring behavior in zebrafish. One is simply looking at the distance moved. I suppose if you're a dizzy fish, you wouldn't move as much, because if moving makes you dizzy, you would stop. And what we see is that there does seem to be a dose response to some extent that cadmium, increasing concentrations of cadmium result in a much different mobility. So that sort of contradicts what I just said. These are actually much more mobile compared to the controls. And interestingly enough, the addition of calcium chloride has no effect on their behavior. Now, it doesn't seem to be a monophasic type of response, because between 40 and 60, we've tested 50, and we don't necessarily see that distance move change very much relative to the control, although the trend is higher. So they do exhibit some increased activity. And then when we look at the dimensions of the otoliths, so we can actually measure these and plot them, we see that there is a significant decrease in the overall size of the otoliths relative to the wild type. And this is actually seen across all but one concentration of cadmium. Now keep in mind that these concentrations of cadmium that we are using are what are considered acceptable concentrations of cadmium in the environment for people to be exposed to. So what is the composition of the otolith? We already told you it's high in calcium carbonate, but the protein matrix consists of several proteins: otopetrin, otoconin, spark, otolin, and slumpy. And then there are various sulfated proteoglycans that make up the entire crystal itself. So naturally we wanted to know, well, can we affect the size distribution of the otoliths in exposed zebrafish by supplementing them with calcium? Perhaps cadmium is interfering with a calcium-dependent process. And we also wanted to test whether any of these proteins, which happen to be very rich, one of the properties of these proteins is they're very rich in protective cysteines, meaning that they are not typically oxidized, even though they are extracellular. So we wanted to know, well, is cadmium increasing the redox state of this otic fluid such that even protected cysteines might be affected? And to do these experiments, we looked at various concentrations of calcium chloride as well as the reactive oxygen species N-acetyl N-acetylcysteine. And what we saw was that addition of calcium had no real significant effect on the size of the otoliths in unexposed zebrafish. But if you look over here, addition of 2.5 millimolar calcium in the presence of 60 parts per billion cadmium rescued what was otherwise a much smaller size in the otoliths, suggesting that this additional calcium is compensating for some suppression of a calcium pathway within the inner ear. On the other hand, the addition of N-acetylcysteine had no effect on it. So calcium rescued the otolith, but the N-acetylcysteine did not. On the other hand, coming back to this hyperactivity figure, where we show that the activity of these zebrafish is significantly increased in the presence of cadmium. When we add calcium, we don't see any rescue. In fact, addition of calcium alone seems to increase the hyperactivity of the fish. But when we add N-acetylcysteine, we don't see any statistically significant difference between the control and the cadmium exposed, but in the presence of not zebrafish. Although it's still higher, so we're working to refine these concentrations. These, incidentally, are not toxic to the zebrafish, so experiments are underway to look and see whether or not we can increase this rescue of behavior by an increased addition of that. And then when we look at the expression profile of the genes that regulate, that encode these proteins, the only one that we see differentially expressed in the presence of cadmium is OTOP1, encoding odopetrin. It is significantly upregulated by 36 hours, even though by 24 hours it's actually downregulated. So there's some kind of a biphasic process going on right here. But what we don't know yet is what the relationship is between the changes in OTOP mRNA and the protein composition of the otoliths. So to do that, we are actively dissecting otoliths. We've gotten very good at this. And in collaboration with Dr. Behrman, we'll be looking at the protein composition of these otoliths to see if there are any changes in both identity of protein and also perhaps oxidation state. We are also interested in identifying the calcium channel, and we have a candidate, an L-type voltage-gated calcium channel. We're looking at the ultrastructure of the otoliths to see if they are significantly different between controlled and exposed. And we're trying to unravel this complex relationship between the redox state, otolith deformities, and behavior to see if there is a connection there. So that's where that work stands at the moment. The last story I'd like to tell you is this cautionary tale on the reliance of morpholinos to understand protein function. So for those of you who are not sure what a morpholino is, here is a phosphodiester bonded ribose base. This is the backbone of DNA. Over here is a phosphodiamidate morpholine backbone. So it kind of looks like that, but not exactly. But the nice thing about it is that you can covalently bind nitrogenous bases to it, just like you can in DNA. And you can use that as an antisense technology to either inhibit splicing by blocking a splice site or inhibit translation by blocking the start site of translation. And therefore either produce a prematurely truncated protein or an RNA that doesn't get translated and is degraded through nonsense RNA-mediated decay, or to simply abolish the protein expression. So morpholinos, up to 1989, from 1940 to 1989, there were only about 300 publications for morpholinos. And a lot of this work was done by Stephanie Denning, a master's student who's now at UNC Chapel Hill, and many of you may recognize her. And over the course of the next 10 years or so, there wasn't really a big increase in morpholino publications. But then came along Steve Ecker's highly cited paper, Effective Targeted Gene Knockdown in Zebrafish, in 2000. And from 2001 to 2016, there were over 5,321 papers in which morpholinos were used in some developmental study or another. So there's a huge exponential growth in the use of morpholinos. Our association with morpholinos came about because we were looking at the effects of dioxin on zebrafish development. It was long known that zebrafish were sensitive to dioxin, and it resulted in craniofacial abnormalities. And so when we did a transcriptomic analysis of exposed zebrafish, we uncovered a battery of genes that are always associated with activation of AHR, but also a number of transcription factors, including FOXQ1 and several other FOX genes. And it's a highly conserved protein within the fork-head box domain among vertebrates. And TCDD selectively upregulates it. By about 24 hours, there is a huge, there's at least a 4-fold induction, up to 8-fold induction by 48 hours. And we know from in situ hybridization where this upregulation occurs. It's limited to the branchial arches, which will give rise to the upper and lower jaw, and then eventually moves somewhat posterior into other branchial arches. So we tried morpholino rescue, thinking that if FOXQ1 is upregulated by TCDD, maybe if we inject FOXQ1 morpholino and then expose them, we won't see this effect. But before we did that, we just tested the morpholino, and lo and behold, we got very similar phenotypes. You've seen this figure before, craniofacial abnormalities that look very similar to what we saw with TCDD. And we thought, well, maybe not enough FOXQ1 is bad, and just as too much FOXQ1 is bad. It's a transcription factor, so there's probably some regulation of its level of protein at the state within the cell. So we went ahead with the experiments, but we were not able to rescue TCDD-exposed zebrafish. But we thought, well, why don't we do a transcriptional analysis of these fish to see if, in fact, we can uncover genes that are regulated by FOXQ1, which in the absence would have changed expression. And we were aware of some concern in the field about the activation of the p53 pathway. So we did a very large experiment where we took a control morpholino that is thought not to have any effect, the wild type, FOXQ1 morpholino in the presence of p53, and FOXQ1 morpholino alone. And for the most part, they clustered together. There is some disparity within the biological replicates, but there was significant clustering by PCA analysis. And when we looked at the changes in gene expression, we were quite surprised to see the difference between wild type versus the FOX-only morpholino compared to wild type versus the FOX plus p53. There was a difference of over 1,000 genes that were misregulated in this subset that were not misregulated in that subset. And that seemed quite surprising to us because p53 alone couldn't possibly be responsible for that. We saw similar issues with the control morpholino, but what we were most concerned about was these 42 genes that seemed to cluster right here. This was common to all these different pairwise comparisons. And when we analyzed those genes, what we saw disturbed us. They were all clustered within the p53 signaling pathway, the ATM signaling pathway, as well as a GAD45 signaling pathway. These are all pathways indicative of stress. And this occurred even with a control morpholino, which has been used as a control throughout all those 5,000 papers that you saw. And it's still nevertheless causing substantial cellular stress. And that cellular stress can be manifested in cell death. So here you see background cell death by acridine orange in wild-type fish. The FOX morpholino causes massive cell death. FOX plus p53 reduces that amount of death, but the control is essentially indistinguishable in terms of numbers from the FOX plus p53. The control alone is causing sufficient cell stress in activating cell death pathways. And we think that a lot of these cells are actually neural crest cells. And if you look closely, it's often possible to see changes in the patterning of the craniofacial elements, which are neural crest cell derivatives, in these control fish. But they don't rise to the level of concern, so everybody sort of ignored them. But clearly there's something going on here. So to get to get something out of this big data set, now we're characterizing a CRISPR allele of FOXQ1b, comparing that to our morpholino data set. We're also trying to identify targets of FOXQ1 transcriptional regulation. And then a brief summary of other things I've done. So the zebrafish core facility was funded by NC Biotech from a grant I received. I've already told you about this, the different capacities. It serves multiple labs, and there's room for more investigators. And I couldn't have done a lot of this with Vicki Martin's help, who helped me to navigate the bureaucracy. I have lots of different collaborations going, most prominently with Carolyn, of course, but lots of different people within the institution, including some educational collaborations with Hien Tran, Jim Gillen, and H.T. Banks. And I'd just like to highlight a few of these. The first is the BD2K project, which was funded. Both David Reif and I are co-eyes on that. And we have a manuscript from that project. The other collaboration is one with Michael Behrman and Catherine Hoyo, where we're doing epigenetic and proteomic profiling of cadmium-exposed zebrafish and submitting a grant in October. With Seth Coleman, we're looking at the analysis of atypical AHR ligands, vitamin D deficiency, and neural function, and we have a manuscript in review right now. And with Heather Patisol, I recently published a paper with her on transcriptome analysis in the Clarity BPA study, and that paper is in endocrinology. And then, as embarrassed as I am, I still have a very large set of RNAs that I need to work up for Jim Gillen with a collaboration that he and Carolyn and I engaged in looking at the brain transcriptome analysis of guppies under different predation regimes. And I promise that I will get to that, Jim. So I've published a few things. This is the collaborative workshop that Jerry talked about. Paper came out of that. This truly is a consensus report. We all got together and we hammered out what we need to do to make zebrafish the premier model for toxicology. And it includes people like Warren Casey, Stephanie Padilla, Robert Tangway. David is in here somewhere, I believe. Right, David?
I'm near the end.
You're near the end, Robert Tangway. So it includes a lot of us here at State as well as at various organizations within the area. As a result of that meeting, John Hamm at NICEDM and I have put together the CSIC Consortium, Systematic Evaluation of the Application of Zebrafish in Toxicology. It includes all these participating organizations, including the military. I don't know why they're interested in that, but they are. And funding for this project we're expecting in mid-2017. I also have a collaboration with Dr. Mamta Bell at NIEHS to analyze TOX21 chemicals for evidence of neurotoxicity in zebrafish, and I received unofficial notice that that project will be funded as well. I teach a lot because that's one of the main reasons I came to State. And the courses that I teach, I think, are the best that we have to offer. They include developmental biology, signal transduction in the environment, and a new bit course I put together on looking at genome editing in the zebrafish. I get a lot of postdocs and graduate students. It would be great to see a faculty or two take that class as well, and then run off with zebrafish and do wonderful things. I couldn't have done a lot of this without all the people who have worked in my lab. Carson Heck is a new graduate student as well, came in with Adrian Green's class, and I didn't have a chance to talk about his work. But these are all the people that have come through my lab at one point or another, and they've all gone off to better things. And I want to extend a special thanks to Chris Cole, who has, Chris Cole. Dave, I was going to say David Cole, Chris. For managing our zebrafish facilities, done a spectacular job, but unfortunately we're losing him. But none of this could have been done without the support of my family, and these are the current members of my family, and hopefully no more will be added. And this is my 6-year-old son, who actually hiked that mountain by himself. Kudos to him. And with that, that's all I have. Question? Yes, Robert.
Oh, I wanted to ask just 2 questions. It was a nice presentation.
Thank you.
First, when you talked about flunomide, yeah. You mentioned that the main risk factor for people is teratogenesis. And I was wondering, how does in terms of when you think of translation, how does the effect on dopamine and mobility in the zebrafish model relate to the carcinogenic effect, which is the main thing that happens in people?
Effect. Yes. So it is contraindicated in pregnancy because it is a teratogen in model organisms. It's been shown to be a teratogen in rabbits, it's a teratogen in zebrafish at high concentrations. So it seems that the prudent thing to do is not take this while you're pregnant. But because leflunomide exists in a precursor to active state, and the precursor, leflunomide itself, is capable of entering the blood-brain barrier, as we showed with our adult zebrafish, we think that that is at least a reason to be cautious about using this drug and to monitor these patients carefully for any signs of neurological problems later on. There does happen to be a link, an epidemiological link between rheumatoid arthritis and Parkinson's disease. And that link has not been explored all that long. So we don't know if that link existed prior to these DMARDs coming on the market, which, like leflunomide, may cross the blood-brain barrier and have other effects, or if these observations were made post introduction of these compounds into the market. So that's why I think that the zebrafish can help uncover some potential risk factors associated with the drug. We're not saying take it off the market, but what we are saying is that maybe this deserves a second look.
Well, I won't use it. And I had one more question.
Dexamethasone.
One more question that's related to the cadmium work and the heavy metal work. So it's quite well known, I think, that many heavy metals, for example, lead exposure, have effects on the central nervous system, especially on like neurotransmission channels. So when you do your study on the zebrafish, is it possible that the effects that you see on activity have absolutely nothing to do with the otoliths, but may be due to effects on the central nervous system? And moreover, is it possible, if the otolith is affected, but it's bilaterally affected in a similar way on both sides of the fish, that they're actually doing fine, unless they start like going on their back or circling or doing weird things which would be obvious. But maybe they're doing fine, and the effects that you see might be due to effects on the CNS.
Right, so I think the second issue that you raise can be resolved by the observation that there are several mutant zebrafish which have abnormally sized, bilaterally symmetrical otoliths, and they still exhibit behavioral disorders. So it's an important component of the behavioral integration system of the fish. The first point that you raised about whether it has nothing to do with the otoliths and is strictly neurological, and we were trying to address with using antioxidants versus cadmium or calcium to see if we could tease this apart. Now, we're not neurologists, so what we are looking for is a relatively simple path to get at the answer without having to become neurobiologists. But if we have to become neurobiologists to address this question, rest at ease, we will become neurobiologists to address this question. But yes, you raise a very good point. I think that given the fact that we can rescue the otoliths with calcium but not the behavior, there is an uncoupling, and we don't deny that. Does that address your question?
Sure.
Okay. Jim?
So back to this drug again, we came in one person.
Yeah, I hope nobody's worried here. I mean,
but you're looking for the melanocytes are missing, or well, you're
Yes.
looking for the melanin, and how do you, are the melanocytes there, or are the neurocrest cells themselves deficient in this?
So that's a good question, and that's one that we don't have a handle on yet. We have markers for neurocrest cells in the undifferentiated state, and we're using those to determine whether or not those neurocrest cells are there, but there's just something in their ability to differentiate. Now, I will say that we do get craniofacial abnormalities associated with exposure to the flutamide at high enough concentrations. So we know that at high enough concentrations, it does adversely affect neural crest cells.
Some neural crest cells may be more sensitive than others.
Absolutely.
But that also goes back to the question of, some of these things aren't developing correctly. Have you looked at those to see if there's a common morphology of
the fish?
Yes.
Common morphology, they are horribly deformed in all sorts of directions. What we have done is we're in the process of raising the resistant fish and then seeing if we can't come up with a crossing speed that would allow us to figure out.
2 separate lines of fish, or is this just in a population?
Within a population, yeah. So it's going to be a complicated process. But we would welcome any interest in collaborating from any of the faculty within the genetics program to help with that. Tree, I'm looking at you.
Sequence them.
I know, that's exactly, yeah, but that requires cash. Yes.
So, sure I understand this right. So the flutamide is converted to an active agent, and that active agent is what's effective in the rheumatoid arthritis.
That's correct.
But only though the flutamide crosses the blood-brain
That's
barrier.
correct. As
So
far as
are the neural effects direct from the flutamide, or is the enzyme present that converts it to this?
That's a good question.
And if it's direct, can they not use the active agent as to treat instead of the flutamide?
Yeah, and so both prescriptions are available. So both are commercially made. And as far as I can tell, and I have scoured the literature for this, I don't know why you would prefer to prescribe the flutamide over teriflutamide, unless there is some absorption issue associated with it, or some intolerance to one versus the other. To address the question about, this is an interesting story. We were at SOT, the Society of Toxicology meeting, and we had a poster up. We think leflunomide causes these neurological deficits in zebrafish. And we got a visitor from pharma. He said, oh, we make that drug. I'm like, oh, you do? It's like, yeah. It's like, would you like some of it that is radio-labeled? He's like, we'd love it, because that would enable us to do all sorts of really interesting experiments that we can't do right now, because we have no way of detecting at the moment where leflunomide is. Well, 1 month turned into 2, turned into 3, turned into 4, and eventually, rather than just saying, well, I'm getting my superior's permission to send it to you, they completely stopped talking to us. Now, I don't know why, but we don't have that compound on hand. And to synthesize a radio-labeled form of leflunomide, probably not something we're going to be able to do.
Do you have a marker for the enzyme? Do you know where the enzyme's expressed?
Well, so yes, and those enzymes tend to be more or less ubiquitously expressed, so it is possible that it is being metabolized into teriflutamide. And the kinetics of that are fairly well known in certain animals like dogs, but not necessarily in people. And so the residence time of this unmetabolized compound within the central nervous system is really not known. Other questions? Yes, John.
If you treat the leflutamide fish with L-dopa, I think I said it right.
Yes.
Leflutamide, okay. If you treat them with L-dopa, you rescue the...
Did we rescue them? Not even marginally, right?
The behavior or the
I
lens?
There
think
was-
it's problems with the oxidation.
Yeah.
We had problems with the oxidation
In the water.
in the water. So if, one thing we thought about was intraventricular injections of L-dopa, because the ventricles are quite large in zebrafish.
Yeah.
Yeah, and just do that. So that's an experiment that we could do.
Yeah. So would you predict that it would rescue both the locomotive behavior and the melanocyte, the melanin production?
We wouldn't expect it to, L-dopa, we wouldn't expect it to rescue the melanocyte.
It should be specific to the locomotive.
That's right.
If it's a pathway that you think.
That's right. Because even though the pathways converge on tyrosine, those pathways are not present in different cell types. So there are some pigmented neurons that make a neuromelanin. So you would expect that pathway to be active there. And I guess that's why the substantia nigra gets its name. But the homologous neurons in zebrafish are not melanated. We wouldn't expect it to have an effect. And the other thing is that, to my knowledge, melanocytes or even neural crest cells do not express the pathway for dopamine synthesis. Jerry, you have a question?
If I recall correctly, we provided evidence that the behavioral effects caused by the flutamide are mediated at least in part by the AHR.
Yes.
Yes. So do you see that type of behavioral effect with dioxin?
Dioxin is so toxic that the fish blow up and they don't move at all. But they don't move because it's physically impossible for them to move. You can watch them under the scope. It's a tragic thing to watch, actually. Their pectoral fins are moving like hell, but they can't get anywhere because they're so diffused and so the answer to your question is there a ligand to AHR that doesn't cause that overt toxicity, which, when administered, could cause those behavioral phenotypes? And I think the answer is that there probably is some evidence of AHR causing central nervous system inflammation, which is why we're interested in that. And that was one of the things that we were going to look at, was the effect of these atypical AHR ligands on neurobiology. So
Could you just titrate down the concentration
of leflunomide?
No, of TCDD.
Oh, TCDD? I don't think there's a safe concentration of TCDD for zebrafish. Russell.
Hey, Tony, why do you think the calcium enhanced the behavioral activity?
So that's a good question. And what we think is, and I have a student actually scouring the protein structure database to identify calcium or cadmium binding sites within proteins. One of the beauties of cadmium is that it's a heavy metal. And as a heavy metal, it scatters X-rays like crazy. People, when they were doing isomorphous replacement to solve crystal structures, would dope the crystals with these heavy metals and diffract them. And if the diffraction space didn't change, you could use the changes in intensity to figure out the protein structure. So by looking at the protein database and looking at all these different channels that have been crystallized, particularly the ectodomain parts of the channels, we're looking for potential associations between cadmium and these channels. But the other reason I think that calcium rescued it is because there is some evidence in the literature that cadmium can interfere with calcium transport. And it probably interferes via inhibiting some of these calcium channels from functioning properly. There is evidence that these L-type voltage-gated channels are present within the epithelium that lines the aortic vesicle, and they're responsible for pumping it. So we're going to look at both agonists and antagonists of those L-type channels to see if, in fact, we can't recapitulate some of the observations we're seeing with cadmium or calcium to try to get a handle on that. John?
So Tony, have you considered whether adding the calcium chloride into the water is altering the driving force for moving calcium across those channels and transporters, right?
So you asking me to solve the Nernst equation?
Well, I mean, if you've increased, right? But if you've increased the amount of calcium in the external fluid,
I'm sure it is.
And the driving force is already strong, but if you've made it a lot stronger, and cadmium is really, maybe you've got an incomplete block of the L types or the transporters, and then the ones that are open, there's more calcium.
That's what that's our hypothesis, is that the increased chemical gradient of the extracellular calcium that we're adding is actually overcoming any inhibitory effect that cadmium might be having on these channels, simply by the kinetics of displacement. Yes?
Yeah, calcium will seriously tighten up the permeability of the gill, and it will slow down cadmium uptake.
Is that right?
And it will also compete with cadmium for binding sites.
Oh, okay. See, I should have talked to you about this
Yeah.
before. So basically what you're saying is that the fish are just not experiencing the same concentrations of cadmium. How can we test that?
We can test that.
We should test that. Jim?
Well, I'll just mention that what in the 70s or 80s anyway, what I understood to be the highest concentration of dioxin in the world is about 2 hours east of here in Plymouth, North Carolina, a place called Welch's Creek that goes into the Roanoke River and was created by a paper mill there that was used in a process called the Kraft process of making pulp that it was chlorinated and made dioxins. So I don't know the situation, you know, whether that supposedly that stuff doesn't degrade very much. But what I thought I'd just mention that that's there, and what I was thinking about was evolution of resistance. That I actually took samples from that when I was an undergraduate, and where the paper mill effluent went into the Welch's Creek, 10 meters up, it was just gorgeous fish communities and nice invertebrates and everything. And then where the water came in, it went anoxic. And nobody knew about dioxin, at least nobody professed any knowledge of it at that time. But it went anoxic, and then it was no fish, but very abundant tubophysids in particular in the sediments. And there'd be some tubophysids up there, but since there are no fish, I think the tubophysids were, you know. So looking at invertebrates through there might, if somebody wanted to, be a local place to look for something like that.
That's an interesting question, because Mark Hahn at Woods Hole has scoured the East Coast, in particular the region around the Chesapeake Bay, where there has been significant sedimentary deposition of dioxin and dioxin-like compounds, and around the Hudson Bay as well, as a result of all the manufacturing that occurred along those rivers. And what he uncovered is that a number of invertebrates had actually acquired resistance to this 2378 TCDD by simply mutating their AHR receptor to no longer be able to bind it. And so even though they have the aerohydrocarbon receptor, they're completely irresponsive to this very potent activator of AHR. So yeah, these selective pressures can lead to relatively quick changes in these invertebrate organisms. It's pretty clear that this phenomenon that he observed is actually due to the selective pressure placed on the species by TCDD because, and I'm not an ecologist, but I guess sympatric organisms just above the region that's contaminated are actually susceptible to AHR. They're the same species, I guess. But one has actually undergone an adaptation-driven event that's absent from the others just within the span of 70 to 80 years, or however long dioxins were being dumped into those rivers as a result of manufacturing. Now, the other thing I will say is that the Kraft process does lead to the production of these compounds, but not all of them are polychlorinated the way TCDD is. So you can have different locations of the chlorines. And so the backbone structure of the dibenzoparadioxin moiety can be chlorinated in 8 total different places. Only 2378 is extremely toxic. The others have varying degrees of toxicity. But you're right, they are persistent. They don't break down very easy. It takes a lot of UV light to break them down. They tend to make it down into the sediment, so if we don't disturb the sediment, they don't leach out very much because they're stuck to other organic material. But yeah, that's a really interesting question from an ecologist standpoint. Other questions? Okay.
Thanks very much, Tony.
Live long and prosper.